Dear student, today we will deal with the pistil, Megasporangium, means ovule, and the development of embryo sac. Let's proceed. First, the pistil. Pistil means that is the unit of gynosium, and gynosium is the female reproductive part of the flower, as we know. And the pistil has three parts. Stigma, style, and ovary. Stigma, style, and ovary. This is stigma, this is a style, and this one is ovary. And within the ovary, ovule means this structure. Stigma is the platform to receive the pollen grains or site of pollen grains. As soon as pollen grain will lie here, pollen tube will germinate after the secretion of stigmatic fluid, which is mainly fructose, and in the development of pollen tube, boron element is playing very important role. So this is the stigma. Stigma may be rounded. Stigma may be cap-like, like this, maybe like ovate, maybe bifid, maybe feathery, like this, and may have such type of hairs due to which the surface of the stigma becomes volvate in texture. Now the style. The style may be short or long. The style has a very narrow capillary tube through which pollen tube passes, and then the first male gamete and second male gamete will come here and fuses. Now this swollen basal part of the pistil is called as ovary. This is the most important part of the gynosium and most important part of the flower, because this ovary has ovule, and within the ovule, embryo sac. This structure is the embryo sac. And within the embryo sac, egg apparatus, central cell, and antipodal cells are present. So now, this is the well-developed structure of anatropous ovule. There are mainly five shapes of the ovule. The first is the orthotropous. This is the very good example of the gymnospermic ovule. The second one is the anatropous ovule. This is the special feature of the angiospermic ovule. This type of ovule, when the chalazal end and micropylar end just lies on the right angle to the funicle, this is called as hemitropous. When the ovule becomes curved, this is the campylotropous. When the shape of the ovule, or mainly embryo sac, this one is the embryo sac. This shape looks like horseshoe shaped, that is the amphilotropous. And when the embryo sac and ovule is just circulated or coiled, that is called as cercinotropous. So there are mainly 5 different shapes of the ovule: 1, 2, 3, 4, 5, and this one is the additional. Sometimes it happens. Now, dear students, this is the well-shaped structure of an anatropous ovule. This is the opening, and this is called as micropyle, through which pollen tube enters. When the pollen tube enter, firstly it will penetrate to the synergid. The number of synergid cells are 2. This is 2 in number. And the feature of the synergid is that before the entry of pollen tube, one synergid will degenerate, and the another synergid cell helps in the entry of pollen tube by the formation of here leafy structure that is called as filiform apparatus. Filiform apparatus. This filiform apparatus, this leafy apparatus helps in the entry of pollen tube. It has been seen that the wall of the egg is one side is the central cell and another side is the synergid cell. So this is the complete structure, is the egg apparatus. Egg is haploid. Egg is haploid as well as the synergid is also haploid. This one is the central cell, that is the big cell, which is binucleate, and these 2 nuclei is called as polar nuclei. So its number is 2. Both are haploid, means N plus N. And opposite to the micropylar end, this end is called as chalazal end. This end has 3 antipodal cells. These 3 antipodal cells are also haploid, means this complete Megasporangium means ovule, and this structure is the embryo sac. This structure is the embryo sac. This one is 7-celled 8-nucleate structure. 7-celled 8-nucleate structure. Now we will discuss about the development of embryo sac, and this discussion comes under the Megasporogenesis. But before the discussion of the Megasporogenesis, there is one point is left here, that is this ovule is the having 2 layering of 2 layered protecting covering, that is called as integument. So this ovule is the bitechnic. If one integument layer is present, that is the unitechnic, and when 2 layers are present, that is the bitechnic. Sometimes a technic ovule is also reported, and within this embryo sac there is the structure which is called as nucellus. We will discuss later. Before the megasporogenesis, let's assume that there is the big potent cells develops within the megasporangium, and this is also called as archisporial cell, and this archisporial cell directly develops as a megaspore mother cell, which is diploid, and this megaspore mother cell will undergo meiosis, and this megaspore mother cell, MMC cell, undergoes meiosis to form 4 cells. This forms 4 cells, 4 megaspore cells. Out of 4, 3 will degenerate and only 1 will be functional. This one is the functional megaspore mother cell. So naturally this is haploid, and this is happening within this embryo sac. So here one potent cell, this is the archisporial cell, which directly behaves like MMC. So this one is the MMC. This is also called as megamyocytes. This MMC, the functional and big MMC, megaspore mother cell, is also called as megamyocyte, which is 2L. It will divide and forming 4 cells here. This is the micropyle end, and this one is the chalager end. 3 will degenerate. The common phenomena is that the 3 will degenerate, and just behind to the micropyle end, 3 will degenerate. And this one means the functional megaspore is projected towards the chalazel end. This type of development is called as polygonum type of development. This is the chalazel end functional megaspore, which is haploid. It's okay? Now let's assume this is the chalazel end, and this one is the functional megaspore, which is potent, having the dense cytoplasm and rich in protein. Dense cytoplasm and rich in protein. So this is the chalager end. So this one is the polygonum type of development. And only one functional megaspore is developing. So it is the monosporic embryo sac. Monosporic, bisporic and tetrasporic. There are the 3 different ways of development of embryo sac. So most common is the monosporic which is also called as polygonum type. If this functional megaspore lies here, means at the micropylar end. Suppose this functional megaspore mother cell was present here. So this is called as Ncert type of development. Micropylar end megaspore. That is the Ncert type of development, and this belongs to the Onagraceae family. Onagraceae family. But generally the chalazal end is functional. This functional megaspore, which is haploid, it will divide by mitosis. Mitosis, cell division. So it will form the two cells. It will divide and forming two cells. Suppose these two cells, two megaspore. Out of two, each one will shift towards opposite, means one will shift towards the chalazal and one will shift towards the micropylar end. One will shift towards the chalazal and another will shift towards the micropylar end. Now this structure it will divide, means again this cell will divide by mitosis. Again these cells both are divided by mitosis. So here four cells. Now again mitosis. So here four cells are formed. Micropyle, this is chalazal end. Now this embryo sac becomes 8-celled, 8-nucleate. This is the embryo sac. 8-celled and 8-nucleate. Again, out of 4 from both sides, one will migrate in the center. As soon as both side or both end will give rise to one nuclei or cell in the center, resulting each end having 3 cells. these two synergid and egg polar nuclei antipodal cells. So here finally an ovule is formed. This is the bitegmic ovule, and this embryo sac is the monosporic embryo sac in which 7-celled and 8 nucleate, because both nuclei or both polar cells will fuse and forms central cell, its number is 1, and this central cell has n plus n condition, and it will exist till double fertilization, and central cell is called as mother cell for the development of primary endosperm nucleus or endosperm. Dear students, in this structure, means in the Megasporangium, each cell has very significant role. To discuss the role of each one, antipodal cell is also very rich in cytoplasm, protein, ascorbic acid, rich in protein, cytoplasm, ascorbic acid, and antipodal cell plays very important role in the development of embryo as well as endosperm. Now, within the embryo sac, nucleus tissues are present, which is also haploid, and this nucleus supplies nourishment this nucleus is also supplying nourishment to the developing embryo. It has been seen that outside of the nucleus there is the another layer. This layer, I mean this one, this is called as endothelial. Endothelial layer. This also provides nutrition and having some fibrous thickening. Now, the integument, if bitegmic integument outer integument will form testa of seed coat, whereas inner integument layer form tegmen. Clear? In case of bitegmic ovule, the outer covering, that is the outer integument forming the testa, and inner integument forming the tegmen of the seed coat. Central cell binucleate, and when double fertilization takes place or going to be taking, both nuclei n plus n nuclei will fuse to form 2n nuclei, and the big central cell fuses with second male nuclei. Or second male gamete which is running within the pollen tube, and finally 2n plus n will form the 3n, and the initial structure is the primary endosperm nucleus, PEN, primary endosperm nucleus. This is the future of the central cell. Then egg. Egg is a very big potent cell. Potent cell, rich in nutrients, and haploid, and this N structure will fuse with first male gamete. First male gamete means that is the N. Haploid structure is an egg and fuse with the first male gamete to form 2n structure. This is called as zygote, and this zygote divide and redivide to form embryo, and in the developing embryo nucellus is supplying the food. Sometimes nucellus may persist, and the persistent nucellus is called as perispun. This is the also unique feature of the nucellus. This one is the synergids. Synergid is having the filiform apparatus, leafy apparatus, which helps in the entry of the pollen tube. This is the basic role of the synergids. And this one is the micropipe through which pollen tube enters. This is the funicle. This is the stalk of ovule. It has been seen that at the junction of the funicle, because funicle attached to the placenta, attaches to placenta, and at the junction of funicle and placenta there is an structure. There is an structure located which is called as obturator. This is also rich in ascorbic acid and protein. The junction of integument and funicle is called as hilum. Junction of integument and funicle. So this is the complete structure of the well-developed Megasporangium, and within the Megasporangium, embryo sac is formed, which is the 7-celled 8-nucleate structure, and the development of Megasporogenesis. That's all.